Hey all, we're going to talk a little bit today about human genetics, how we inherit traits, and how we predict inheritance patterns based upon what we know about genes. This information can be somewhat complex, and so I'm going to move a little bit more slowly than I have in the earlier recorded videos. And there's also quite a bit of information on each of the slides. So I have opted to leave my face off of the slides to make room for all of the information on them. We're going to start off by defining some terms that I will use throughout the PowerPoint. So you're going to be responsible for knowing these definitions, but also for knowing how to use each of the terms that we're going to define initially. So first off, we're going to start with chromosomes and genes. And remember that each of us has 23 pairs of chromosomes. One of each pair came from mom and the other from dad. The word we use to describe this is homologous. We have homologous pairs of chromosomes, and they're homologous because each of the chromosomes in the pair carries the information for the same traits. So in other words, each chromosome carries the same genes. Genes are segments of DNA on chromosomes that code for specific products, most often proteins. It's possible for there to be more than one version of each gene. We call these different gene versions alleles. An allele can be an alternative version of a gene that's located at the same place on the homologous chromosomes. For example, if we look at the gene for eye color, it's possible that mom chromosome and dad chromosome have the same alleles. In other words, the same versions of that eye color gene, which might be the gene for blue eyes. It's also possible that mom and dad's chromosome that carry the gene for eye color have different alleles or different versions of that eye color gene. For example, mom's carrying the gene for blue eyes and dad's carrying the allele for brown eyes. In fact, it's possible for there to be more than just two versions of each gene. When we look at the alleles for eye color, we see that there's the possibility of brown eyes, blue eyes, green eyes, or gray eyes. Those are all different alleles of the eye color gene. Similarly, there are different alleles for the hair color gene: blonde, red, brown, or black. The terms that I've used on this slide and the previous include chromosome, gene, homologous chromosome and homologous gene, and allele. Additional terms are on this slide. Phenotype and genotype are two terms that are used to describe the way somebody looks and the genes that they carry that contribute to that appearance, respectively. The genotype is the combination of alleles that a person carries, and the phenotype is the way that genotype looks on the outside. If an individual has two of the same alleles, we say that that individual is homozygous. If, on the other hand, an individual is carrying two different alleles for the same gene, we say that that individual is heterozygous. Alleles can be either dominant or recessive. an allele is dominant if it overshadows the appearance of the other. An allele is said to be recessive if it is able to be overshadowed by another version. On this table from your textbook, we see that when we look at the two different versions of alleles for freckles, we see that freckles are dominant to not freckles. An individual who is heterozygous for freckles would still have freckles. The only way an individual would have not freckles is if he or she were homozygous for the recessive allele. The terms that I've used on this slide include genotype, phenotype, homozygous and heterozygous, and dominant and recessive. Now let's use the terms that I've defined. Because chromosomes and the alleles on them distribute independent from one another during the production of sex cells, the combination of alleles in sex cells is almost infinite. And I want to look at two genes, each on different chromosomes, to exemplify that infinity. We're going to look at the genes for hair color and eye color, for example, and we're going to assume that the gene for hair color is on the larger chromosome and the gene for eye color is on the smaller chromosome. We're going to also assume, for simplicity's sake, that each of these genes only has two different versions. The gene for hair color being only blonde or brown, and the gene for eye color being only brown or blue. We're going to assume that the pink chromosome, which came from mom, has the allele for brown hair on the big chromosome and the allele for brown eyes on the little chromosome. We're going to also assume that the big blue chromosome, which came from dad, has the allele for blonde hair, and the little blue chromosome, also from dad, has the allele for blue eyes. So mom is carrying the gene for brown hair and brown eyes, and dad is carrying the allele for blue eyes. When we produce sex cells, we do so via a special type of cell division that's called meiosis. When we were talking about the generation of sex cells in the reproduction part of this class, we said that it was a two-step process, or it required two cell divisions. The first of those cell divisions was quote-unquote normal because the DNA was copied, and the second division was not normal because chromosomes were divided into daughter cells or distributed into daughter cells without cell division. That's sort of true and sort of not true. In this process of meiosis, it's true that the first cell division is normal in that the DNA is copied, but it's abnormal or different from mitosis, which is the type of cell division that occurs in not sex cells. It is abnormal because of the way chromosomes are distributed. The two copies of mom are distributed into one daughter cell, and the two copies of dad are distributed into the other daughter cell. In the process of mitosis, each of the daughter cells gets one copy of mom and one copy of dad. So mitosis differs from meiosis in that the chromosomes are distributed differently. When we look at that second row, after the chromosomes have been distributed in meiosis 1, we see that the daughter cells could have either mom's big chromosome and mom's little chromosome, or dad's big chromosome and dad's little chromosome, or they could have mom's big chromosome and dad's little chromosome, or dad's big chromosome and mom's little chromosome. So there are 4 different possibilities. After the second round of cell division, when the duplicate chromosomes are separated into daughter cells, it generates 4 different possibilities for sex cells. Either they're carrying the alleles for brown hair and brown eyes, or they're carrying the alleles for blonde hair and blue eyes, or they're carrying the alleles for brown hair and blue eyes, or they're carrying the alleles for blonde hair and brown eyes. So 4 different possibilities. In this example, we are looking at 2 genes on 2 chromosomes, each of which have 2 allelic variations. In the human genome, we have roughly 25,000 genes, each with a variety of allelic variations on 23 different chromosomes. So when we consider all of that variability, we can see that it is indeed true that sex cells have almost infinite possibilities for the alleles and the combination of alleles they carry. Despite that array of infinite possibilities, it is possible to predict inheritance, and in fact very easily, if we look at only one gene at a time. We're going to do that by using freckles as an example. There are only 2 versions of the freckle allele, either the version that produces freckles or the version that produces not freckles. If we look at a mating between a female who has freckles and an individual or a male who has not freckles, we can predict what their offspring will be like. We're going to assume that mom is homozygous for freckles. Of course, we know that dad is homozygous for not freckles because that is the only way he could not have freckles. The other thing that you have to know is that freckles is dominant over not freckles. When we look at mom's production of eggs, when her freckle gene distributes into the daughter cells, because she only carries the freckle allele or the freckle version of this gene, all of her eggs will carry that freckle encoding allele. Similarly, because dad only carries the allele for not freckles on his freckle gene, his sperm can only carry the not freckle allele. When mom's egg and dad's sperm meet at fertilization, the zygote will have one of mom's freckle alleles and one of dad's not freckle alleles. In other words, the embryo will be heterozygous, and that embryo will develop into an individual who will have freckles because freckles is dominant to not freckles. We can predict the likelihood of offspring having freckles using something called a Punnett square. In this case, dad has no freckles, so he is homozygous recessive, and mom is homozygous dominant, having freckles. Dad can only produce sperm that carry the not freckle allele, and mom can only produce eggs that carry the freckle allele. All of their offspring will be heterozygous for freckles, and all of them will have freckles. If two parents are homozygous for different alleles of the same gene, in this case freckles, 100% of their offspring will be heterozygous for freckles. Because freckles is dominant, 100% of their offspring will be phenotypically freckled. We're going to make things a little more complex now by assuming that we have heterozygous individuals. On this image on the right part of your slide, you see what it would look like, what sperm production would look like in a male who is heterozygous for freckles. He received from mom the gene for freckles and the gene from dad for not freckles. When this new dad is producing sperm, the alleles for freckles will distribute differently such that half of his sperm will carry the allele for freckles and the other half of his sperm will carry the allele for not freckles. In this case, dad can produce sperm that carry the freckle allele or the not freckle allele. If if mom is in the same heterozygous genetic state, her eggs will similarly be half carrying the freckle allele and the other half carrying the not freckle allele. If we use a Punnett square to predict what the offspring will be genotypically and phenotypically, we see that 1/4 of the offspring will be genotypically homozygous for the freckle allele. Half of the offspring will be genotypically heterozygous for the freckle allele, and 1/4 of the offspring will be genotypically homozygous for the not freckle allele. So, two heterozygous parents will produce 25% of their offspring that will be homozygous for one of the alleles. 50% of their offspring will be heterozygous, and 25% will be homozygous for the other allele. The genotypic ratio then is 1 to 2 to 1. The phenotypic ratio, however, is 3/4 of their offspring will be freckled, and only 1/4 of their offspring will not have freckles. Therefore, the phenotypic ratio is 3 to 1. These ratios are then used to predict what the likelihood of these two parents producing a freckled offspring would be. In other words, in this case, 75% likelihood that their offspring will have freckles, but there is a 25% likelihood that their offspring would not have freckles. Let's give this a go. We're going to predict the genotypic and phenotypic ratios of a mating between a heterozygous mom, because freckles are dominant, she has freckles, and a homozygous recessive dad without freckles. The first step is predicting what this sex cells would look like. Mom, because she's heterozygous, has 50% of her eggs that carry the freckle allele, and 50% of her eggs carry the not freckle allele. Dad, because he doesn't have freckles, can only give the not freckle allele. We're going to transfer that information from the allele that the sperm and the egg are carrying to our Punnett square. And we see that half of the offspring are heterozygous and therefore have freckles. Half of the offspring are homozygous for not freckles and therefore have no freckles. In this case, there is a 1-to-1 genotypic ratio where half of the offspring are heterozygous and half of the offspring are homozygous. And the phenotypic ratio is also 1-to-1, where half of the offspring will have freckles and half of the offspring will have not freckles. Other traits besides freckles are encoded by a single gene. Those are shown here. A few of the traits are shown in the images. For example, widow's peak is where your hair sort of forms a Dracula-like peak on your forehead. Unattached versus attached earlobes, or the ability to roll your tongue. Those are all single gene traits. Other single gene traits are whether or not you have a straight thumb or a bent thumb, sometimes referred to as a hitchhiker thumb, a straight pinky finger or a bent pinky finger, or a cleft in your chin, a dimple in your chin. You can Google what these might look like, but these are all single gene traits. We can use something that's called a pedigree chart, shown here, to determine whether a trait is dominant or recessive by looking at the way it's inherited over several generations. A pedigree chart is shown on the left, and it shows a mating between a male, the square shape, and a female, the round shape. In this case, the female, because it's shaded blue, happens to have the trait, and the male, because it's not shaded, happens to not have the trait. This male and female produce 6 offspring. Half of their offspring, number 1s, 2, and 3, carry the trait, and the other half, numbers 4, 5, and 6, don't have the trait. Offspring number 1 and offspring number 3 get married, and they mate to produce offspring. Offspring number 1 is a male who happens to have the trait, and he marries and mates with a female who doesn't have the trait. They produce 1 offspring that has the trait. Offspring number 3, also a male, mates with a female who doesn't have the trait. They produce 3 offspring, 2 of which don't have the trait, and 1 of which does. Based upon the presence of 3 offspring in the second generation that have the trait, and 3 offspring that don't have the trait, half with the trait, half that don't, we can predict that mom is a heterozygous individual. And we can do that because 2 slides ago, we did a mating like this, where we saw that half of the offspring carried a trait and half of the offspring didn't carry the trait. This enables us to predict that mom is carrying a dominant trait or a dominant allele, as well as a recessive allele. In this pedigree chart, This is what inheritance would look like for a recessive trait. Mom and dad mate, and they don't display whatever trait we're thinking about. So they produce a single offspring, a female, who also doesn't display the trait. She mates with a male who doesn't display the trait, and they have 4 offspring, 2 of which have the trait. How is that possible? Well, it must be that mom and dad, the original mom and dad, are heterozygous, producing their daughter in the second generation, who is also heterozygous. She's heterozygous. She marries or mates with a male who is heterozygous, and they produce half of their offspring that are homozygous recessive. You can do a Punnett square to show how this would happen. Up until now, we've been talking about the inheritance and inheritance patterns and our ability to predict that for one gene at a time. But it is possible for us to predict inheritance patterns for multiple genes in combination. It certainly is more complex, however, if we do that. Probabilities for genes can be determined in combination using the same technique, a Punnett square, that we've used in previous slides. However, when we're looking at the generation of gametes, we have to look at all of the possibilities of those combination genes. In this case, we are mating 2 individuals who are heterozygous for 2 different genes, one for freckles and one for widow's peak. When we look at the possibility of gamete production, we see that dad and mom each can produce 4 different possibilities. 1/4 of them freckle widow peak, 1/4 of them freckle straight hairline, 1/4 of them no freckle widow peak, and 1/4 of them no freckle straight hairline. The Punnett square has 16 different mini squares in it, and each of those 16 squares tells us the genotype of the predicted offspring. Knowing the dominance and recessive nature of each of these 2 traits, we can predict what their offspring would look like. Indeed, 9 out of the 16 will have freckles and a widow's peak. 3 of the 16 will have freckles and a straight hairline. 3 of the 16 will have no freckles and a widow's peak. And only 1 of the 16 will be completely recessive and have no freckles and a straight hairline. That is a typical 9:3 to 3:1 phenotypic ratio. Now remember, this is looking at only 2 genes with 2 different alleles, and humans have 25,000 genes, each with the possibility of having more than just 2 different allelic variations. So while it's possible to do Punnett squares for 3 and 4 different gene combinations, they get very complex. It's also more complex when we consider genes on the X chromosome. One example of a gene carried on the X chromosome is the gene for a blood clotting enzyme. If this gene is non-functional, it can result in a disease called hemophilia, where individuals are unable to clot blood properly. So they have a lot of bruising and they bleed very easily. Genes like this blood clotting gene on the X chromosome are inherited in a gender-specific manner. We say that these are sex-linked traits. Sex-linked traits are generally carried on the X chromosome, and this is a diagram of the X chromosome showing you that it does in fact carry or encode for a number of different genes. Because females have 2 X's and males have only 1, it's possible for recessive traits to be overshadowed by a dominant version of the allele in females, but it is not possible in males because they don't have a second allele to overshadow that recessive trait. When we look at the production, or when we look at a Punnett square for a sex-linked trait like hemophilia, the father will produce either X chromosome carrying sperm or Y chromosome carrying sperm. Of course, moms, because they have 2 X's, will produce eggs that are carrying X chromosomes. When an X chromosome carrying egg meets an X chromosome carrying sperm, girls are produced. When an X chromosome carrying egg meets a Y carrying sperm, boys are produced. If the X chromosome is carrying the normal blood clotting enzyme gene, of course, the girl that is produced will be genotypically homozygous and normal. Boys that are produced will be carrying the X chromosome with the normal blood clotting gene and a Y, so not a homologous gene. If the X chromosome that is mutated is distributed to an egg and that meets an X chromosome carrying sperm, a girl will be produced who is heterozygous for the blood clotting allele, but will be phenotypically normal. Unfortunately, if that same egg meets a Y carrying sperm, there is not a homologous allele contributed by the Y carrying sperm. So the non-functional blood clotting enzyme will be phenotypically visible. In other words, this boy will have hemophilia. Finally, we've been talking about traits as if all of them were encoded by a single gene, and that's simply not true. The vast majority of traits, including height, skin tone, body shape, face shape, you name it, are controlled by many different genes. When a trait is controlled by many different genes, we say that that trait is polygenic. And again, polygenic means that more than one gene contributes to that phenotype or that trait.